It's time to get started. Hello, everyone. Greetings of the day. On behalf of the BioProtocol webinars team, this is Antara Sengupta, welcoming you all to our webinar series titled New Tools of the Trade in Life Sciences, Technologies that Enable Breakthroughs. For those joining us for the first time, BioProtocol is an open access, peer-reviewed online journal dedicated to making research more transparent and reproducible. We sincerely thank you all for joining us from different parts of the world and being part of our mission. Today, we will learn about zapERtrap, a light-regulated ER release system to study secretory protein trafficking from our speaker, Dr. Ashley Burke. Dr. Burke, thank you for accepting our invitation. We have our BioProtocol webinars program lead, Dr. Renee Weisbauer, as the moderator for today's session. Before we start, I would like to share a couple of quick announcements. If you have any questions related to the webinar topic, you can post them in the Q&A and chat box throughout the talk. Our speaker will address them at the end of the presentation. The webinar will be recorded and posted later on the website as well as on our YouTube channel. So if you wish to revisit it, you can do so. And lastly, if you would like to receive a certificate of attendance, we encourage you to participate till the end of the live webinar. So with that, Renee, I hand it over to you now. Thank you.
Thank you so much. Okay. All right. It is my distinct pleasure to introduce today's webinar speaker, Dr. Ashley Burke, who is currently a postdoctoral fellow in Dr. Aaron Truman's lab at the Max Planck Institute for Brain Research in Frankfurt, Germany. Dr. Burke completed her PhD in pharmacology in Dr. Matt Kennedy's lab at the University of Colorado Anschutz Medical Campus, where she studied how proteins are being delivered with high spatiotemporal precision within neuronal dendrites. And during her PhD, she also developed a method for controlling secretory protein trafficking using light, which she will talk about today. She's currently utilizing advanced mass spectrometry-based techniques in her current research to study ribosome heterogeneity and specialization in neurons. Ashley is also dedicated to promoting diversity and equity in science, serving as an equal opportunity officer at her institute and as a secretary of Natives in Neuro, a global organization supporting indigenous researchers in neuroscience, as well as participating in women in STEM initiatives. And with this, Ashley, I am already very much looking forward to your talk, and the floor is all yours.
Great. So thank you for that wonderful introduction. I'm really grateful to be here. And I'm happy about this opportunity to share some of the work I did during my PhD, and that was published a few years ago now. And so my hope is that this webinar helps to answer any questions you have about potentially utilizing zapERtrap, or with my, Midwestern accent, I call it zapERtrap, in your research. And so I'll start off with an outline for today's webinar. So I'll briefly discuss a little bit about the history and existing methods that allow the synchronous ER release of cargo proteins from the ER, which of course have broad applications in understanding secretion dynamics, studying vesicle trafficking, and dissecting regulatory mechanisms within the secretory pathway. I'll of course introduce the tools, so zapERtrap, Zapalog-endoplasmic reticulum trap, and kind of, go through what motivated us to develop this tool. I'll show some proof of concept experiments in fibroblast cells, and then, of course, go to my favorite cell type, the neuron, and show just some of the work that we did that was previously published a few years ago, where we were able to study compartmentalized secretory trafficking using our tool because we were able to locally initiate ER release from subcellular regions. I'll go over some considerations and limitations of the tool. Okay, so now coming to our sort of basic cell biology overview. Hopefully you are already aware that nearly all integral membrane and secreted proteins are processed through the cellular secretory network. So in eukaryotic cells, these proteins are synthesized in the ER, where they are folded, assembled, and they undergo various post-translational modifications, including glycosylation and disulfide bond formation. And following maturation, proteins leave the ER at specialized exit sites and accumulate in the Golgi apparatus, where they are further biochemically modified and ultimately sorted into mobile carrier vesicles that shuttle them to the appropriate subcellular domain. So different protein cargoes utilize distinct trafficking routes, and some of these routes are still poorly characterized. So this highlights the importance of new tools and approaches for investigating secretory protein trafficking. So classical experiments tracking proteins as they navigate the secretory network were done by fractionation of cells and newly synthesized proteins that were pulse labeled with radioactive amino acids. Now, this method is not ideal because it requires cell lysis and solubilization. So it therefore limits the information that you can obtain about the spatial organization of secretory trafficking. This, of course, changed in the mid-1990s after the advent of genetically encoded fluorescent protein tags. So if you tag your protein of interest with a fluorescent protein and you conditionally sequester it in the ER, you can simply watch it as it traffics out of the ER into different subcellular compartments in real time using time-lapse microscopy, and therefore ask, where does this target protein end up in the cell? And so one of the first efforts to directly visualize a protein on its journey through the secretory network in live cells utilized a fluorescently tagged version of a temperature-sensitive mutant of VSVG that is retained in the ER at elevated temperatures, but then traffics normally when cells are cooled. So by simply decreasing the temperature, one could release VSVG from the ER and visualize its progression through the secretory network. And this powerful approach provided the first glimpse into the cellular organization of protein secretion, yet the major drawback was that it wasn't generalizable to different proteins and it lacked, fine temporal control. And so to address these issues, several different approaches emerged that use small molecules rather than a temperature shift to trigger forward trafficking of proteins in cells and diverse model organisms. And so the first chemical inducible release system, sort of schematized here, involves a self-associating mutant of FK506 binding protein, or FKBP. And so when tandem copies are fused to a target protein, they cluster the protein and they prevent it from leaving the ER. And then by adding an analog of rapamycin known as Rapalog, these clusters rapidly dissociate and the proteins that were trapped in the ER can then continue on their way through the secretory pathway. Another approach is the rush system retention using selective hooks. And this system involves adding biotin to release SBP tagged proteins from an ER localized version of streptavidin. So by adding biotin, you're basically competing off the SBP tagged protein. More recent systems are so-called protease controlled secretion systems, which rely on the proteolytic cleavage of an ER retention signal, such as like a KDEL motif, after reconstitution of a split protease, so activation of a split protease by addition of a small molecule, and those are different depending on the system. And so these trap and release strategies are really powerful for studying secretory trafficking, especially in diverse cell types, but they lack the spatial regulation required for precision control at the level of individual cells or even subcellular domains. And so one could, maybe try perfusing the ligand, but of course, a less technically challenging way to trigger local ER release would be to use light. And so the first and only light-inducible ER release system, apart from zapERtrap, is based on UVRA, which is a plant photoreceptor that forms photolabile homodimers. And so the strategy was also developed by the Kennedy Lab, so where I did my PhD and where zapERtrap was developed. And it uses UVB light to trigger the release of UVR8 tagged proteins from the ER. Now, it sounds like this is all well and good, but the system does come with some drawbacks. So first, UVR8 is not a small protein. It's actually almost 450 amino acids. And I think, in general, the bulkier the tag, the greater the likelihood that that tag could interfere with the localization of the target protein. Secondly, it requires UVR8 to be in the cytosol, not in the ER lumen, which is a downside if you want to surface label your target protein in the end. And then lastly, UVB excitation light is challenging to focus for local activation with conventional optics. And so overall, these existing methods are not really well suited for local ER release. And the ability to manipulate the surface levels of specific receptors or secreted proteins within user-defined cell populations would be a powerful strategy for studying cell signaling, especially in intact organisms. And then furthermore, to come to my favorite cell, in large cells of the nervous system, such as neurons or glia, whose secretory organelles are broadly dispersed, the ability to initiate secretory trafficking from specific subcellular domains, such as dendrites, axons, or the cell body, would address questions about how or whether proteins are spatially compartmentalized in complex cellular environments. And so there was, of course, still a need for tools that enable control of secretory trafficking in both time and space. And so finally, this is where zapERtrap comes in. And so our approach uses a small molecule called Zapalog that was initially developed by Tom Schwartz's lab at Harvard. And it consists of two major moieties that are connected to each other by a photocleavable linker. And so one is a synthetic ligand of FKBP, abbreviated here as SLF, and the other is the antibiotic trimethoprim, which binds really tightly to bacterial DHFR, or dihydrofolate reductase. And so in this way, Zapalog essentially dimerizes these two protein domains, FKBP and DHFR, and you can rapidly disrupt their association by cleaving apart Zapalog with 405 nanometer light. And so to use Zapalog for light-triggered ER release, we fuse DHFR to a secretory cargo, as shown here, and we transfect cells with this construct along with an ER localized or ER targeted version of FKBP. And importantly, we add Zapalog at the time of transfection so that when the DHFR fused cargo is synthesized in the ER, it can be efficiently trapped there. And then upon brief illumination with low intensity 405 nanometer light, when you shine that on your region of interest, the link between the cargo molecule and the ER anchor is rapidly broken, and then forward trafficking of the cargo can proceed. And so what does this actually look like? And so here I'm showing you the light-controlled release of GFP tag transferrin receptor from the ER of a cultured rat hippocampal neuron. And so transferrin receptor is a generic secretory cargo. It's a type 2 transmembrane protein. And the red dot in the upper right-hand corner signifies the first frame after full field illumination. And right away, all these vesicular structures begin to appear in the dendrites. And you can see this just massive redistribution of TFR to the somatic Golgi. And so the next thing that we needed was a really sensitive way for detecting where this released cargo was showing up on the cell surface. And so after trying a few different strategies, the one that worked the best was simply performing these experiments in the presence of dye-labeled extracellular antibodies, which can rapidly bind a surface epitope that's presented when the released proteins finally reach the plasma membrane. And so to demonstrate this, and just see how this works, here's sort of a proof of principle experiment using Cas7 cells expressing, again, GFP tag transferrin receptor that's initially in the ER. It's initially retained there. And then you see all of these, white puncta beginning to appear soon after ER release. And it's because we have the dye-labeled GFP antibody, present in the imaging media, and they're basically binding and essentially cross-linking the transferrin receptor as it's being inserted into the plasma membrane. And so using this real-time surface labeling strategy coupled with our zapERtrap system, we can characterize the complete trafficking itinerary for a given secretory cargo. And so, for example, as you might expect, we saw that the onset of transferrin receptor surface accumulation corresponds to a time, after, post-release, where accumulation in the Golgi is already at its peak, because TFR is, first trafficking to the Golgi and then to the surface. Another proof of concept is that we can trigger ER release within a specified region. So here we steered the beam of 4 or 5 nanometer light over just this Cas7 cell. And you can see robust Golgi accumulation and subsequent surface accumulation only in the targeted cell. And so collectively, these data indicate that our zapERtrap system can be used to rapidly, robustly, and locally trigger the forward trafficking of proteins using a single pulse of light. Another thing that I do want to also mention here is that another benefit of our system is that, having light be the trigger is beneficial and advantageous because then it offers you gain control. So you can essentially dictate, how much you want to release by, illuminating with lower intensity light and so on. Okay, so how does this actually look in neurons? And can we target subcellular regions? And of course, the neuron is a wonderful cell to try that out. So before kind of diving in, I just want to give a little bit more context as to why local ER release in neurons would be really impactful. And for that, I kind of have to take a little detour and just really briefly, my computer's a little laggy, but I just need to briefly mention the spatial organization of the secretory pathway in neurons. If we look at the organization of the secretory pathway in neurons, we see that the ER is in almost every compartment, including axons and the tips of dendrites. And there's also rough ER out in the dendrites, and even often at the base of dendritic spines, as you see in this electron micrograph. So this is just indicating that maybe there's local synthesis of integral membrane proteins, and presumably those that are important for synaptic function. And so even if you're not, a neurobiologist, I don't think you really need to know so much just to really appreciate just, how spatially complex and puzzling the secretory pathway is, because the Golgi apparatus is found in about 20% of dendrites, and it's largely confined to the cell body and to the proximal region of the apical dendrite. And so this has raised a really big question in the field as to why bother making integral membrane proteins distally out in the dendrites if you don't have all of the appropriate organelles to actually process them and traffic them locally to the cell surface. And so using our tool, we set out to ask the simple question, what is the spatial relationship between where a protein exits the ER and where it actually shows up on the cell surface? And how might this be influenced by neuronal activity? So the first thing we did was to test whether neuroligin 1 and GluA1, which are both postsynaptic receptors that are important for synapse function, can they be efficiently released from the neuronal ER after a single 50 millisecond pulse of light? And so similar to transferrin receptor, which is shown here on the left, neuroligin 1 robustly trafficked out of the ER and redistributed to the Golgi within minutes. GluA1, on the other hand, so GluA1 is a subunit of the AMPA receptor, which is really important for synapse function, this protein took about twice as long to reach peak accumulation in the Golgi. However, this is actually consistent with previous studies demonstrating the relatively slow ER export of AMPA receptors. And so after confirming that we could efficiently control forward trafficking of these synaptic cargoes using our system, we applied the same real-time surface labeling strategy to determine where and when they showed up on the cell surface. And we developed just an automated process that identifies distinct surface cargo accumulations, and it essentially measures how far away they are from the cell body. And so here I'm showing you heat maps that show the time that each surface cargo accumulation first appeared. And we call it surface cargo accumulation because we can't say if it's actually just a single molecule. It's likely not. But these heat maps are showing kind of the timing of when the surface cargo accumulations first appeared following ER release. And also it shows you where it appeared on the cell. And so we found there to be differences between the two cargoes, neuroligin 1 and GluA1, in terms of timing of surface delivery in the distal dendritic compartment, as you can see here. And an easier way to look at this is to compare the time it took for each cargo to reach 10% of the max surface accumulation within these three compartments. So the cell body, the proximal dendrites, or the distal dendrites. And from this, you can see that neuroligin 1 surface delivery in distal dendrites occurs at significantly later time points than GluA1, even though it traffics faster to the Golgi after ER release, which was really interesting. And so overall, these results suggest that spatial temporal trafficking properties, even after just full field illumination, are cargo specific. And a major feature of zapERtrap is that it allows local ER release from different subcellular compartments. And that's, of course, why we engineered this system in the first place. And so we, of course, wanted to look at that. And so we leveraged, this feature to ask whether protein cargoes exiting the ER within specific regions of the dendritic arbor are selectively delivered to the nearby plasma membrane, or are they dispersed as they move through mobile downstream trafficking organelles. And so this is just sort of a proof of principle where we can illuminate here in this segment of dendrite that's outlined by the purple or pink box. And then we can also take a look at, an adjacent control dendrite. And so what hopefully you can see is that there are these punctate structures that are forming within tens of seconds in the illuminated dendrite, but not in that adjacent dendrite, nor in the cell body. So this is really confirming precise, dendrite-specific ER exit selectively within the illuminated region. And now if we kind of take a look at an actual time-lapse movie of this, what you can see, if I play this again, is that following local dendritic ER release, we see these vesicles appearing and they're rapidly moving and they're being transported out of what we call the release zone. So essentially the region that we illuminated. And so I'm not going to, of course, go into all of the details here since this work's already published, but using this tool, we were able to find that proteins that enter the dendritic secretory pathway are actually rapidly dispersed before surface insertion. And this was an unexpected finding. We were thinking, we were sort of assuming that we would see perhaps surface accumulation occurring, nearby the release zone, the area of release. Another question, that we want to address with our system is to ask whether cell body derived proteins can be directly trafficked to the surface of dendrites through long range vesicular trafficking, such as, through vesicular trafficking with molecular motors, on microtubules, and so on. And so to address this, we used this time focal illumination to release either neuroligin 1 or GluA1 from the somatic ER. So instead of focally illuminating dendrites, we're illuminating the cell body. And we basically ask, do they appear on the surface of the cell body, the soma, the dendrites, or both? And we also layered on some activity and asked whether neuronal activity played any role. And so following local somatic ER release, cargo accumulated in the somatic Golgi with no detectable accumulation in vesicular or Golgi-like structures in either proximal or distal dendrites. So again, confirming that cargo's released from this time the soma, the somatic ER network, are primarily utilizing the somatic secretory network rather than diffusing to dendritic ER exit sites. And in the minutes following somatic ER release, both neuroligin 1 and GluA1, so here's neuroligin 1 and GluA1, appeared at the cell surface at the soma, the cell body, as well as tens to hundreds of micrometers into the dendritic arbor, which, yeah, this was faster than we had expected. And then elevating neuronal activity with this drug known as bicuculin, the details aren't super important here. But what we found is that the total amount of GluA1 that was delivered to the cell surface was increased, but elevating activity did not have an effect on timing of surface delivery. So it didn't increase the timing of surface delivery of GluA1. But actually the converse is true for neuroligin 1. So really showing some cargo-specific effects and trafficking routes. So elevating activity in the case of neuroligin 1 did not have an effect on the total amount of surface delivery, but it actually impacted the timing of surface delivery. And so there was detectable neuroligin 1 appearing sooner at distal dendrites. Okay, so I, of course, am not going through every single detail or figure of this paper, but I did want to finish going through this application in neurons with what I think is a really cool finding, and a finding that I don't think we could have, reached without this tool. And so using this tool, we noticed that for some cargoes, for neuroligin 1 and transferrin receptor, there seemed to be this robust accumulation of the cargo at what's known as the axon initial segment. And it's really interesting because, all three of the cargoes that I've talked about today are somatodendritic cargoes. And what I mean by that is that they don't localize to the axon. And so, yeah, this to us was intriguing, and we were able to kind of more carefully look at this by also co-expressing a marker of the axon initial segment, and that's called incrin G, so that we could, definitively, look and see that those cargoes were appearing at the AIS surface. And here I'm just showing how that's not the case for all cargoes. So for GluA1, we did not see this AIS accumulation. And so this is just representative images showing sort of before ER release and then at select times post-release. And the top is neuroligin 1 and the bottom row is GluA1. And yeah, you can kind of, it's quite clear that for neuroligin 1, we see this kind of interesting finding. And so here, this is just quantified across several cells where we calculated the fraction of the total surface, cargo, that was, localized to the AIS. And importantly, we can, of course, characterize the kinetics of this accumulation and how that it compares to surface delivery at other subcellular regions, such as the cell body and the proximal and distal dendrites, like I mentioned before. But what's really important, and I think this is a control that one really needs to consider when wanting to study secretory trafficking, is, are we seeing this phenomena because we're just simply kind of overloading the secretory pathway? And so what we did here is we could actually precisely titrate the amount of neuroligin 1 that's released from the ER simply by decreasing the intensity of the excitation light. And so what we did is so this is actually showing you the intensity of neuroligin 1 in the Golgi. So we could calibrate the amount of neuroligin 1 release by quantifying first its accumulation in the Golgi following excitation with different light intensities. And what's important here is that we observed, strong AIS delivery even at 405 nanometer excitation intensities that are near the threshold for reliably detecting surface trafficking. So this was, suggesting to us that this was an actual, biologically meaningful observation and not just, a caveat of our tool. And another thing that I think this nicely shows is that we observed very little GLUA1 signal at the AIS, even when we use saturating light intensities that release the maximum level of GLUA1 from the ER. So just further evidence. Okay, so I want to talk a little bit about the construct design and plasmid design. So currently, the status with zapERtrap is that it is an overexpression system, and I'll show you, I think on the next slide, some of our attempts at making this such that it's a knock-in construct. But for now, I'll kind of go over what we have and what we already have published. It's in this methods molecular biology chapter, where we also outline details of how to transfect this in fibroblast cells and neurons and all of the details for that. Okay, so coming to the plasmid design. So there's two main components as I've already introduced. So we have the ER anchor, and then we have, of course, the cargo construct. And so there's some, of course, essential components of the FKBP anchor construct. So, of course, first and foremost, we need an N-terminal signal peptide sequence. And here, the sequence that we have used is just the mouse immunoglobulin kappa light chain. And this is for proper ER targeting. And then you might want to include an epitope tag to just confirm expression and ER localization of your anchor. And then we also have a C-terminal KDEL sequence to actually retain it within the ER lumen. One thing that you might consider is to utilize tandem copies of FKBP to enhance ER retention of your DHFR fused protein. And we've tested this and we haven't seen any indication of these tandem copies affecting the kinetics of cargo release. But of course, we've only tested that in a handful of cargos, so this would have to be empirically determined. You can also utilize a fluorescent protein instead of an epitope tag if you would like. For cargo retention, there's way more considerations here. So first, DHFR is fused to the terminus that's within the ER lumen. So you have to ask yourself, okay, what is the membrane topology of my protein? So in order for DHFR to be available for Zapalog-mediated binding to the anchor, it has to be positioned in the ER lumen. And so what I just have kind of schematized here is whether you have, let's say, a type 1 transmembrane protein like neuroligin 1 and GLUA1, or a type 2 transmembrane protein like transferrin receptor. And so DHFR, you would want to fuse that on the N terminus in the case of a type 1, on the C-terminus in the case of a type 2. So another consideration is if your target protein already contains an internal ER targeting sequence. So you, of course, need to have some sort of signal peptide. Some proteins already have an internal ER targeting sequence, so that's something that you would definitely have to know, because otherwise you would have to include it, and you would definitely include it upstream of DHFR. Or upstream of, your cargo. Third, perhaps you would want to include an epitope or a fluorescent protein tag, or maybe even a protease cleavage site, or, for example, thrombin. And that's actually what we've done in our studies. So that's just a way to, in case you have a little bit of leakiness, you can sort of adjust for that. And then lastly, something to consider is using a bicistronic construct. And so if you want bicistronic expression, so utilizing maybe an iris construct, you can do so. This might actually help you achieve more uniform expression ratios across cells. And then you don't also have to like co-transfect multiple plasmids. And yeah, I guess the major thing to consider here is to prioritize the expression of the anchor. And so for that, positioning it upstream of the iris sequence. Okay, so adapting zapERtrap for your experiment. So I have listed here just some considerations and limitations. This isn't, of course, an exhaustive list, just some of the things that I've been thinking about. So the KDEL ER retention module may not be suitable for all eukaryotes. So I, of course, am biased to neurons and other mammalian cells. So it may be that if your cell of choice, your organism of choice doesn't actually have KDEL receptors, then you would want to utilize a different ER retention motif. Another thing is to consider permeability of Zapalog. So Zapalog needs to be able to get into cells. It needs to get into the ER lumen. And so delivery of Zapalog to cells might be confounded in the case of the C. elegans cuticle. So I don't know if anyone has tested this yet, but that would be something to consider. Also the cost. So Zapalog currently is not very cheap. So it's around 1,500 euros for just 5 milligrams. Now a little goes a long way. And we've actually even been successful at reusing it. So there are ways to kind of cut costs in this case. And then sort of like I mentioned earlier, I think the biggest caveat, and as with, almost all of these inducible release systems is, it requires protein overexpression. And this is tricky because we're studying the localization and the trafficking of proteins, but, if we're modifying that in any way, then we're not actually, studying what we think we are. And so these were just some of my attempts, 4 years ago when I was in the lab at, basically having a CRISPR knock-in of DHFR GFP to the endogenous locus of the NR2B subunit of the NMDA receptor. And so this was very successful. I had only tried it out a few times with the FKBP anchor. And so, yeah, more optimization would have to be done there, but it is possible. Okay, so lastly, of course, there are 7 different zapERtrap constructs on AdGene. So you can, feel free to use. And with that, I'd like to thank the people who helped with this project, and really my PhD mentor, Matt Kennedy, whose, idea this really was, and Erin Bowen for doing some of the early experiments, Sam Schwartz for her help with image analysis, and then Dean, Mason, and Christina, who did additional experiments for the revised manuscript many years ago now, 3 years ago now. And then also, of course, thanks to our collaborators, Amos Gutnick and Tom Schwartz, for providing the Zapalog reagent. And so with that, I'm open to take any questions. And of course, feel free to reach out if you have any questions as well.
That was a beautiful talk. Thank you so much, Ashley. There are a few questions. Sashin, I hope I'm pronouncing that right, asked, can you tell us a little more about DHFR and FKBP, and maybe in the sense of how big they are? Are you using domains? Are you using the full proteins? Have they been modified in order to recognize the Zapalog specifically, so that it doesn't bind to other FKBPs, for example?
Okay. So
if that is the
to my
case.
knowledge,
That's
yeah,
a lot of questions.
so
I'm sorry.
no, it's great. So we are using the full length. So DHFR, let me see.
Approximately.
DHFR is around 21 kilodaltons, so 187 amino acids. And then FKBP is, yes, smaller. It's 12 kilodaltons, around 108 amino acids. And so definitely smaller than, let's say, the UVR8 base system. We're, like I said, we're using the full length protein for each case. And to my knowledge, they're not modified in any way. So especially for dihydrofolate reductase, trimethoprim, which is, what's binding to DHFR, is an antibiotic. So normally binds to bacterial DHFR. And then in terms of FKBP and binding to the SLF, yeah, it's not modified in any way. It's not specialized in any way.
Okay, thank you. I really like the Alexa 647 anti-GFP system because it really nicely highlights each of the secretion events occurring and integration into the extracellular solution. I have a couple of questions. One is, so it's immobilized, right? Once it's entering that space. Is that by forming a cluster and just it being so big, or how does it get immobilized?
Yeah, so it's just based on the essentially the cross-linking by the antibody. And we've done experiments actually both at the AIS and then just also over the entire cell, just to really show that it's surface, that it's remains at the cell surface. And so what we can do with that is we can wash off, let's say it's Alexa 647 GFP. We can wash that off, and then if the GFP is rabbit, then we can apply an Alexa 568, anti-rabbit secondary, and just really be able to show that it's on the cell surface. It's not like that accumulation, somehow accessed the surface and then was endocytosed and then went somewhere else. Yeah. So it's just cross-linking with the antibodies.
Really cool. And it really helps amplifying the signal and then probably enables easier quantification. I was wondering if let's say for some organisms that system would not be easy because of penetration into an organism, would it be possible to observe insertions using a fluorescent protein, for example, linked to the cargo without that signal amplification?
Oh,
Does that make sense? I'm not.
Yeah, so I just think it'll be diffuse because then it'll diffuse. So I think your question is, if you don't have that antibody binding, but you want to see it reach the cell surface. So that was the beautiful thing with the antibody, is that it would cross-link and presumably, although we can't say with definitive certainty, that it's showing the locale of surface insertion too. So if it's just a fluorescent protein, there's nothing that's going to keep it kind of tethered and immobilized there. So it'll then be able to diffuse along, throughout the plasma membrane. And actually, to just kind of getting at your first question, in the manuscript and one of the supplementals, we showed the stability of these surface accumulations as well. And so it's just, one example, but you can see some of them appear. And then, if we continue, of course, imaging and tracking, you can see that those same surface accumulations remain, and some of them kind of grow a little bit over time as well.
Ruby Wong asks, wonderful talk, Ashley. I really enjoyed seeing all the beautiful movies and how well your technique works. Axonal ER has been bubbling to be a key player. Have you tried photoactivating in the axon or the AIS? And would the secreted protein have a more localized pattern after ER release in the axon compared to the dendrites?
Okay, yeah. So thanks, Sophie. I tried photoactivating the axon of A's. I have not. Specifically, no. I was very much focused on dendrites, and of course postsynaptic compartments, as you probably already know, are kind of my focus even now in my postdoc. So no, did I try? No, I did try. I made a vGlut1 version of this, and I had trouble with leakiness, so I didn't pursue that any further, but that was something that we did want to do and we did want to try, of course. But yeah, so short answer is no. And then would the secreted proteins have a more localized pattern after ER release in the axon compared to the dendrite? Yeah, so yeah, I don't know because we didn't try it, but so when you say localized pattern after ER release, that perhaps, with surface delivery, if we see it kind of closer to that region of release, I mean, I would assume just because, in the axon, it doesn't have perhaps as much, places where it can actually go. So yeah, it's something that someone needs to try out.
Gershwin Zias, again, I hope I am pronouncing that right, said, excellent work. What were some of the challenges you faced during the study?
Oh, so yeah, so some challenges. So as I sort of alluded to with leakiness, so that's why we engineered a thrombin cleavage site between DHFR and, DHFR, our epitope, then we had our thrombin cleavage site and then our cargo, so that we could sort of cleave off, any existing cargo that was on the cell surface already. We also had trouble, I think some of this actually written down because it's been some time now. Right, so initially we had the FKBP on the cargo and DHFR on the anchor, and that didn't work very well. So when we swapped those domains, then it worked much better. Some other things, some things that we've tried that, didn't make it into the manuscript, and so co-release of different cargo molecules from the ER in the same neuron to kind of compare trafficking itineraries in the same cell. So we tried this a little bit, but definitely needs more follow-up. Also, too, being able to visualize the proteins as they move through the secretory system. So so, they are relatively acidic, in oxidizing environments. And so there are FPs that are more oxidation resistant. And actually one of those is included on AdGene that, showed a little bit finer detail and ability to see those vesicles kind of transit intracellularly.
Cool. Thank you so much. Well, there's a lot of thank yous and nice talks in the chat. Everyone really enjoyed this presentation, and so did I. So thank you again.